Okay, hopefully that is recording. I will just start the slides right now. Hi everyone. Welcome to the Biology 3 and 4 lecture for this holiday lecture series. I hope all of you have been having a good holiday. Hopefully some revision's been taking place. If not, today is going to be a revision. If this is your first lecture in the series, welcome to ATAR Notes. If this is your second, third lecture in the series, we're happy to have you coming back. Just before we get started, I have a couple of housekeeping rules for you guys today. You will be watching the stream as it is for the next 2 hours. However, if you do have any questions of any type throughout the duration of the lecture, there is a chat box down that you can type your question into and I'll be able to give you a response. It will be me answering all of them. So if you have any questions about bio just in general, or QCE or uni life just in general, outside of the content of the lecture, please feel free to answer. I can try and give you a response to the best of my ability. So yeah, welcome to ATAR Notes. And so we are, just a little bit of a background about us. We're a company that was created by high school students for current high school students. So all of the tutors who are working, at least in the Queensland branch, we're all ATAR graduates, and our whole purpose is to be making resources that make your life a little bit easier. We are now 3 years into the ATAR system, which is a much better position than it was when I was going through. I was a graduate from 2021. And we're just trying to give you the tips and tricks that we sort of wish we knew, all of the info that we have collected, as well as access to syllabus specific content for biology and any of the other subjects. So if you have not already checked out the resources that we have, allow me to tell you some more. We've got a ton of free resources that are available to you all year round. If you check out our website, which I highly recommend you do, there'll be free study notes that you can download. Sometimes, if you're lucky, you can even get practice exams off there. I know I did back when I was in high school. Every single holiday, we have the free lecture series. So we do them for the main core subjects. And the next lecture series will be in September, focusing specifically on external prep. So if you're in grade 12 and you want to get some questions that are, directly related to the external, that is a place to be. There's also discussion and forum pages, as well as our newsletter, where you can ask any questions that you have about the QCE content, and one of our tutors will be able to reply to you. There are online revision videos and articles targeting all of the QCE subjects and the syllabus and specifics. And also, if you've had a chance to interact with it, there is an ATAR calculator on there. That is based on the past 3 years of scaling results that we've accumulated. So it'll give you like a rough idea of what kind of marks you need for certain ATARs that you maybe have goals for and things like that. Those are the resources that are free to you. However, we do have some more limited edition resources available, created by the company, of course. The first one is our tutoring program. So we do offer private tutoring and class tutoring. All of it is online, all of it is pretty well accessible, and all of our tutors are ATAR QCE graduates now. So you'll be getting the most updated information that you possibly can have, and you'll have guidance by people who have gone through the exact same assessment types. For our written resources, all of our tutors have created a bunch of study guides, including course notes to the syllabus, topic test booklets with practice questions, text guides for all of your English and literature novels. I remember when I was in 11 and 12, I was watching this lecture series along with you guys, and one of the books that I purchased from the 800 Notes bookshop was the Macbeth text guide, because that was a novel that we were doing in English. Super, super helpful, and some of the analysis points in that text guide I actually ended up using in my external essay, and I did really well on that. So I think if you are interested in getting any of our resources, the text guide is definitely the one to get. It's so helpful. I would show it to you guys if I had it on me, but I've given it to a family friend. And it was like notes everywhere and so good. As well as online text guides through the form of Ed Unlimited. So if you um, have watched these lectures before, usually sometimes I will screen share to Ed Unlimited and show you what it is, but it's basically a database of all of our publications. So there's practice exam papers on there, all of our course guide booklets from, NSW, QCE, VCE, all of our topic test booklets for your perusal. It's an online subscription, so you do get access to everything. You can annotate on it. You can um, take like little notes and bookmarks on certain pages that you might want to flag. And it's really, really worth the value. So if you are interested in any of those resources, pop down in the chat and I can give you more details. But for the remainder of the recording, we're going to be focusing on biology. So usually I reckon if I can gauge your pace into the syllabus, you should be entering about mid-unit 4 by now. Most of you would have either completed or would have pending the IA3 or the research investigation. Hopefully, marks went well with that, or it's going well for those who are still completing it. But as soon as that IA3 goes in, you've got a bit of a break period before kind of tackling externals. So this lecture is basically, it's going to be a content heavy lecture, but I have sprinkled in a couple of practice exam questions for you guys. And so I'm going to go through as many exam strategies as I possibly can. And hopefully, everything will make sense. As always, if you have any questions, please let me know. The whole point of these lectures is for you guys to get a sort of taste of all of the content that you're going to be learning within the next couple of weeks. It is going to be pretty full on and it is going to be pretty, I want to say, fast paced. So if you have any queries at all, please let me know. And I will try my best to help with your understanding. We've got a pretty full-on lecture today. So I know it says DNA, genes, and continuity of life, but I'm kind of going to be focusing on the latter half of Unit 4, Topic 1. So that's inheritance and biotechnology. And then on the first sort of third of Unit 4, Topic 2. So that's the evolution topic. Because of that, we're going to be looking at quite a few graphs. We're going to be looking at quite a few processes as well with the biotech section. So please, as always, if you've got any questions, let me know. Other than that, sit tight and we're going to get on with the content. So what you see on the screen right now are all of the cognitive verbs from the syllabus. The QCEE syllabus is available. It's a huge document that's available on the QCEE website. If you scroll down to where it says Unit 3 and Unit 4, you'll come across these massive tables, each with all of the subtopics, and then underneath there are all of these dot points. Those are the exact dot points that you're going to be assessed on in your external exam. And so every time I teach my students or I go through content with any of my students for bio, I'm teaching them according to these dot points because this is basically what the QCEE is saying. This is the info that you need to know. Accompanied with each dot point, so I've got all of the 3 topics laid out in the slide right now. Accompanied with each dot point are these things called cognitive verbs. So you can see at the start of each of the dot point, there is a verb that sort of dictates how they're going to ask you the questions in the external. And each of these cognitive verbs has a different meaning that you need to know to be able to answer the question properly. So some of them are really, really simple. So recognize is just you being able to list out what they're asking you to do. Define. Now, definitions are available to you in the QCEE bio glossary. It's at the very, very end of the syllabus. And it's basically got like the QCEE version of the definition that they expect you to know. Sorry, just give me 2 seconds. I'm going to plug in my laptop so it's not going to die during the recording of the lecture. Yeah, so There are a couple of defined terms in the syllabus. If you have done genetics already, gene and genome are definitions that you need to know. You can get the exact definition that is required off that glossary. I would put them into like study cards or a Quizlet and then just run through them until you've memorized them. Um, Determine and interpret usually involve you reading data off a particular table or graph to find the answer. Describe, so we've got a couple of describes littered throughout the um, syllabus, and that just basically refers to anytime we've got a process. So if you've done unit 4 already, you would have learnt DNA replication and protein synthesis. They are multi-step processes that are pretty important for biology. You being able to describe those processes is knowing each of the steps and like each of the molecules that gets affected in those steps, and it's the same thing for the biotech topic. Predicting frequencies, this is the first thing that we're going to do in today's lesson, is using data about genotypes and phenotypes to make some assumptions or conclusions about future generations of offspring. So we've got a lot of content to get through. I hope you are all ready. Before that, though, let's take a little bit of a break. I want to give you a bit of a background about me. So my name is Amandi, first of all, nice to meet you all. I am a 2021 graduate, so I've been out in second year uni at the moment, and I graduated from Ipswich Girls Grammar School. I studied a mixture of two languages, two sciences, and two maths, so English and Japanese, bio and chemistry, and methods and specialist. Currently, I am a student at UQ studying a Bachelor of Science, and my major is biomedical science. And I also do have provisional entry into medicine. So, as I said before, if you've got questions about the med application process, I can answer that. I know anybody who is interested in medicine is probably looking at sitting the UCAT sometime soon. So good luck to anybody who is. If you have any questions related to content, bio, ATA, please feel free to ask. For bio in grade 12, my raw score for the subject was 98 out of 100. And I received an ATAR of 99.40. So I've been through the process, and I've also spent the last 2 years tutoring biology. I like to think that I know the syllabus pretty well, but QCEE is a little bit of a wild card when it comes to this subject, and we'll talk about that later. But if it is any comfort, I was in your position back in high school. I watched the holiday lecture series in both grade 11 and 12 by ATA Notes, and that's how I started working at the company. So they were helpful for me. This lecture will be like available as a recording to all of you who sorry, if you ever want to refer back to it in your own time, you do have access to the recording, and you should also have access to the lecture slides. At the end of the lecture, I have a little QR code on the slides that you can scan, and it'll give you a transcript of all of the info that I say that may not be on the PowerPoint slides. So you can use that as well to sort of, I don't know, guide your notes, interact with it if you want. And yeah, I hope it's helpful. All right. By the book, I'm kind of just going to be covering everything that you need to know for your external. So because inheritance involves quite a few calculations, I'm just going to turn my laptop around so I can write on the screen. But if any of you have done, actually, wait, sorry. Just so you're not staring at a white ceiling, I'll turn the camera off and then I'll use my laser pointer to show you any main things that you need to know. There we go. Topic 1, inheritance. Okay. If you think back to doing grade 10 genetics way back when, you would have come across these things called Punnett squares and pedigree charts. Essentially, this topic is just a revision of those two concepts and extending your knowledge on them a little bit. And so I'm just going to go through a basic overview of genetics, and then we'll get into all of the inheritance patterns. So a couple things to go over first. The first thing that you guys need to know is what an allele is. So within the human genome, every single one of our cells has 23 pairs of chromosomes. 23 pairs of chromosomes, or 46 in total. If you've seen a picture of a karyotype, you would know what they look like, but essentially, this could be chromosome number 1, this could be chromosome number 2 of the 46, and then the pair of them together is pair number 1 of the 23. One of these chromosomes is a maternal chromosome, so you've inherited it from your mother. The second chromosome is inherited from your father. And the way that it works is each chromosome in the pair is the same version of each chromosome. It's the same version in the sense that every single gene is located in the same spot. For example, this gene that is highlighted here, gene number B, could be the gene for, I don't know, hair color, as an example. And this is the hair color gene that you've inherited from your mother. The hair color gene that you inherit from your father is on the exact same spot and position as the corresponding chromosome. And basically, even though these genes code for a particular trait, there are different variations of the same gene, which are called alleles. So alleles are variations of the same gene. Hopefully, this is revision from grade 10. Same gene. And if we're looking at the basics, they can either be dominant or recessive. And basically, the way that that works is if you inherit a copy of the dominant gene from your parents, that is the gene that gets expressed physically. The recessive gene will only be expressed if you inherit 2 copies of a recessive gene from your parents. The combination of alleles that you may have make up your genotype. So genotype is the combination of alleles and are denoted by letters, basically. So in this case, the gene for hair color, I am marking it by the letter B. Capital stands for the dominant gene. The lowercase b stands for the recessive gene. If you've inherited 2 copies of the same gene, for example, here and here, both of them are the same letter, we say that the genotype is homozygous, this one being homozygous dominant, and this one being homozygous recessive. And if you've inherited a copy of 2 different types of genes, so 1 copy of the dom and 1 copy of the recessive, you are heterozygous for the gene. The alleles making up the genotype will then influence what is known as the phenotype. So this is actually the physical representation of these genes, or the physical trait that the genes code for. Code for. So for example, if you've got a genotype of capital B, where the dominant gene codes for black hair and the recessive gene codes for blonde hair. These are completely fake examples, by the way. Blonde hair. If you've got this is a homozygous dominant phenotype. Dominant genotype, but the phenotype is black hair. Similarly, if you've got capital B, little b as your genotype, even though you do have that recessive allele, the dominant one is the one that's expressed in the phenotype, your phenotype is still going to be black hair. And so all of our inheritance patterns that we're going to be looking at deal with how genes are passed on between parents and their offspring. So if I can go to next slide. Punnett squares are going to be your best friend in this case. They are used to predict the offspring with a parent's genotype and their probabilities. So I'll just go through an example question. This should be revision, but in dogs, there is a hereditary deafness caused by the recessive gene little d. A kennel owner has a male dog that she wants to use for breeding purposes if possible. The dog is able to hear. So the dominant phenotype or the dominant gene of capital D means that they have normal hearing. Since the dog can hear, this dog will either have capital D, little d as a genotype or capital D as a genotype. What are the two possible genotypes of Gilbert? These two. So anytime they have the dominant trait, they can either be homozygous or heterozygous dominant. There. So you can use those Punnett squares to predict what the genotypes of offspring are going to be, or work out what they are using something called a pedigree chart. So what you see on the screen is indeed a pedigree chart. And a couple rules on how they work. The first is that squares are for males, circles are for females. And if one of these parts of the pedigree chart is shaded in, that just means that the person is a carrier of a disease. So pedigree charts are used to map how certain disease and certain conditions can be carried throughout a family. And there are a couple of different inheritance patterns that we're going to learn about that you could be tested on. The first one is autosomal dominant inheritance, as you can tell by the title. Autosomal referring to the non-sex chromosomes, so chromosomes 1 to 22. Dominant meaning that the disease phenotype happens if you have any copy of the dominant gene. So if you've got BB or B, capital B's in your genotype. So let's just say an example of this is Huntington's disease, right? It's a disease that causes nerve breakdown over time, but you're not required to know any of that. Usually, if we have a look at our first parent, or like the starting parent of the family, we've got a male who is affected by the disease. So they could have either been capital B, or capital B, little b, and a female who isn't affected by the disease. So they're double recessive. And the way that we can figure out what the male is like truly is through doing a Punnett square. And so, sorry, just give me 2 seconds. In order to do a Punnett square, you put one of the parents' phenotypes around the top and one of the parents' phenotypes on the side. Draw a little grid like this, and all of the offspring genotypes are the combinations of the alleles that fall into the grid. So this grid square has a capital letter lowercase. This grid square has lowercase, lowercase. This grid square, capital lowercase. This one, lowercase, lowercase. So if that male was heterozygous, the offspring would be expected to be 50% affected or heterozygous as well, and 50% unaffected or double recessive. What we actually see happening is of the 5 kids that are born, 1, 2, 3 of them are affected and 2 of them are unaffected. So it is pretty close to the expected ratio. If they had 6 kids, maybe it would have evened out to 50%. We don't know. But if we take the alternate option and say that the father was homozygous, I'll just do the Punnett square down here. Homozygous with the recessive mother, and then draw the grid. Every single offspring would be capital B, little b, capital B, little b, capital B, little b, or capital B, little b. 100% affected. But that is not equal to the actual pedigree chart ratios. So therefore, we can say that the father is indeed heterozygous and not homozygous. And that is an example of how you use Punnett squares to figure out genotypes. So autosomal dominant inheritance means that the dominant allele gets the disease. However, there is another kind of inheritance that is autosomal recessive. So this time around, the diseased individual or the affected individual must contain the homozygous recessive genotype. Now, in this case, every single black square or black shaded shape on the pedigree chart, we can immediately say is double recessive. It's the unaffected individuals that we are a little bit more wary of. So in recessive inheritance, any kind of recessive inheritance, there are these things called carriers that can start coming up, where the parent is unaffected, but they give birth to a child who is affected. When this happens, you have to realize that the parent is automatically heterozygous. Both parents must automatically be heterozygous. And I'll show you why. Heterozygous. So let's just say our gene this time is like R. This parent could be double R capital. They could also be homozygous. Sorry. I'll go RR, RR, and RR for the recessives. If the parents are both heterozygous, and I do a cross with them, the first offspring phenotype is double R. Then I have two offspring who could be heterozygous, but still dominant, and then 25% chance that the recessive allele gets through. However, if I have a double R situation, and I cross it with, say, heterozygous, because one of them has both copies of the dominant gene, every single one of the offspring will inherit the dominant gene. So none of them will be affected. So the moment that you see this kind of pattern happening where two parents are clean of the disease and they give birth to a child who is born with the disease, that should be like alarm bells ringing that the inheritance type is recessive and that both of the parents have heterozygous alleles. So here, for example, I've got two unaffected parents, one affected child. So this is heterozygous and this is heterozygous as well. Now that I've kind of gone through the whole Punnett square business, the other types of inheritance patterns I'm kind of going to whiz through a little bit. Let me know if you would like further clarification though. Now that we've talked about like autosomal dominance inheritance, we're going to talk about sex-linked inheritance now. This is basically where the gene is on the sex chromosome instead of being in a non-sex chromosome. So X-linked inheritance, because every person is born with at least one X chromosome, this affects everyone. And when you're writing the genotypes of these guys, females will be written as X with like either a capital letter as the subscript or X with a recessive letter as the subscript, or and males will be written as X with a capital or lowercase letter, and then Y to say that they have the Y chromosome there. So in X-linked dominance inheritance, an affected individual has the capital letter, has the dominant gene. The recessive gene is unaffected, and then the Y has no effect. So in this case, for example, the mother would be X capital H or X little h, alternatively X capital H, X capital H. And the father is X little h, Y, unaffected with the Y chromosome. And so you can do Punnett squares to figure out what the rest of their genotypes are. If you have a mother who is affected, who gives birth to unaffected children, that is letting you know that the mother is heterozygous. So she still has a copy of that dominant gene that she can pass. Sorry, she still has a copy of the recessive gene that she can pass on to her kids. And paired with the recessive gene on the dad side, some of the kids will be born with no copy of the dominant gene. Whereas if the mother had both copies of the dominant gene, I'll show you what happens. If I've got XH, XH, and then X little h, Y, every single one of the offspring is given a copy of the dominant gene. and therefore their offspring will all be shaded in. But that's not the case here. So we can immediately say that the mother is X big H, X little h, and Punnett squares can be used to find what the remaining genotypes are. X-linked inheritance can also be recessive, so that just means that the affected condition is the recessive allele. And that any mother with X little h, X little h, so homozygous for the recessive allele, or X little hy, a male with a copy of the recessive allele, will be affected. Typically, for X-linked recessive inheritance, males will have the disease more often than females. And the reason that happens is females have the ability to code against the recessive alleles twice. They have 2 copies of the X chromosome, and they only need 1 copy of the dominant allele to be healthy or born without the condition. Whereas for males, they only have 1 chance to code against the diseased allele, and if they do get it, they have to be born with the condition because the Y chromosome has no effect at all. So typically speaking, in X-linked recessive inheritance, the amount of affected individuals, so all of these guys are X little hy, 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 and then this would be X little h, X little h, X little h, X little h, and X little h, X little h. The amount of affected individuals who are male are going to be much higher than the amount of affected individuals who are female. And unfortunately, that's just the way that it is. An example of this is red-green color blindness and also hemophilia. Generally speaking, males are much more the chance of a male developing this disease is much higher than the females. So as example, I'm just going to use this family right here. 2 unaffected individuals. So the male has to be X capital HY. They don't have the recessive diseased gene. The female could be X capital H, X capital H, or X capital H, X little h, and be a carrier for the condition. Immediately, because one of the boys that are born to the parents are born with the disease, the mother has to be a carrier. So if I was to choose the heterozygous genotype, the mother has X capital H, X little h. The father has X big H, Y. And if I do a Punnett square, I get X capital H, X capital H, X capital H, X little h. So 50% of the females 100% of the females are unaffected, thanks to the father. Whereas the males that are born, only 50% of the males will have the dominant gene, and the resulting 50% have the recessive gene. So in this case, the only offspring that can be born with the disease are males with the recessive gene. And you can work out the rest of those genotypes using the Punnett squares. There is another type of inheritance. Obviously, we've done sex-linked inheritance on the X chromosome, but there is also sex-linked inheritance on the Y chromosome. But all of these diseases only affect males because females do not have the Y chromosome in general. So disease only affects people with XY chromosomes. Now, because of this, the only shapes that are going to be shaded in are these squares. And so let's say that this Y-linked inheritance, the recessive allele, is the one that is the disease. I'm only going to give you one example of this because it's not a very commonly assessed question, but the theory is there. So this father would be X little by crossed with X big B, X big B, or X big B, X little b. So here I have all of the males that are born. So X big B, Y, X big B. I'll start with the heterozygous phenotype first. Every single one of the males that are born has the disease gene, and the other combination of the mother's capital B's. Alright, so here you can see that the, oh, sorry, wait. What am I saying? The disease is on the Y gene, so it's X, Y, little v, and then this is just XX. Now, here we can see that every single one of the sons of the father gets that Y gene inherited. So immediately the disease goes to the sons, but the daughters are spared because it's not on the Y gene. So this would be X, Y, little b. This would be XX. These would both be XX, XX. Now this is XY, the healthy copy of the gene. So in this case, it would be the dominant gene. Here's X, Y, little b, XX. Here, because the healthy copy of the gene is the only Y copy of the gene in the entire family, both of the males are born with the healthy copy. And here, both of the males are born with the diseased allele. Like so. This is an XX, and this would be XY, little b. Y-linked traits are actually very, very rare. Since the Y chromosome only has 63 genes in general. The way that it works is that if the father has the disease, then any sons that are born to those parents will inherit that condition. Right. So those are the main inheritance linkages. We're going to talk a little bit now about things that are called multiple allele inheritance. In some traits, there are multiple dominant alleles. And the inheritance patterns that result from multiple dominant alleles, one of them is called incomplete dominance, where if an individual inherits both dominant alleles, inherits more than one dominant allele, a new phenotype is expressed. So, for example, if you have a cat species that has a dominant allele for white fur, this is dominant, and a dominant allele for brown fur. Oh, wait, actually, I'll go dominant allele for black fur. This is also dominant. If I was to want a cat with only white fur, they would need the genotype CWCW. This is pure white fur. If I wanted a cat with pure black fur, it would be CBCB. But if I was to get the heterozygous phenotype of CWCB, instead of being, white or black, because both of them are dominant, a new phenotype occurs and the fur color would be gray. Completely different to the phenotypes of the homozygous genotypes. So here, for example, checkered box on a pedigree chart means that they've got both copies of the dominant allele. So this could be CWCB. The mother is also CWCB. And if I do a Punnett square for both of these and cross them, one of the offspring will be born one quarter of the offspring will be born as white cats. One quarter of the offspring 50% of the offspring, because there's two of them, will be born as grey cats, and 1/4 of the offspring will be born as black cats, even though both parents started out being grey. And so the whole thing with incomplete dominance is that because there are multiple dominant alleles, a new phenotype occurs when they are heterozygous. Sometimes people say that it's like a blend of effects in the different alleles. And so another example is if you've got a dominant allele for white petals in flowers mixed with a dominant allele for red petals in flowers. Even though it's a heterozygous phenotype, the flower color will be pink, so like a mixture of white and red. So yeah. There is a very similar inheritance pattern to incomplete dominance called codominance. Now, an example of this is blood type, and this was actually assessed in the 2021 paper. So in my external exam, there was a question in paper 2 exactly on blood type. And basically, this has multiple dominant alleles, but also a recessive allele situation as well. Blood types refer to your blood cell's ability to have these proteins on their surface. A-type people have a specific protein on their blood cells. B-type people have a different protein, and then AB people have a combination of the A protein and the B protein. Here, blood types for AB, the alleles for A and B are dominant, but the alleles for the O-type blood are recessive. So we give it like a lowercase I shape. And if the person has both copies of the dominant allele, so if their genotype is IAIB, instead of it being like blood type C, it is a blood type that combines the two alleles together. So you get the protein for A and the protein for B both expressed. A new protein is not expressed. If there was a different protein, then it would be incomplete dominance. But in codominance inheritance, both of the dominant genotypes end up being represented in the phenotype. So anybody with a blood type A can have the genotype IAIA. They can be homozygous for it, or blood type IA little I and be a carrier for the O-type blood. If you have two parents that are both AB blood types and you cross them together, 25% of your kids are going to be type A only. 50% of your kids are going to be type AB. And 25% chance your kids are going to be type B only. And using Punnett squares, you can track what blood types might occur from different kinds of people. And there are some questions that we can look at later on in the lesson that show exactly that. If you want a visual of the difference between codominance inheritance and incomplete dominance, so I've got two flowers here where the parents are white and red. In incomplete dominance, if they get a combination of both of these dominant alleles, the offspring flower will be a blend of the two, so pink. Red and white blend together to make pink. In codominance inheritance, the two dominant alleles share the phenotype, and so there are parts that are red and parts that are white. Hopefully that makes sense. Now, going on to the final topic for the inheritance concept, there is one other multiple allele inheritance pattern called polygenic inheritance, and that is where one characteristic is controlled by two or more genes. This I've given you as the QCEE definition, by the way. And an example of this is human height, that should have a T on there, or skin colors. Skin color actually is made up of 3 genes. So there's a dominant gene, A, B, and C, and then recessive genes, little a, b, and c. The more dominant genes you have, the darker your skin tone is, and the more recessive you have, the more light it is. And so this is actually the example of a Punnett square for inheritance with 3 different types of genes. All of the parent genotypes could be one copy of each of the genes. So either they could be having you could inherit all of the um, dominant genes from your mother and father, or you could inherit all of the recessive genes, or any combination of, two dominant genes and one recessive, or two recessive and one dominant. And basically, once you do a Punnett square, for example, if I was to get the genotype of this individual, you would need to combine the alleles from one of the parents with the other parent, and you would have capital A, capital B, little c, capital C, little c. So they have equal amounts of recessive and dominant here. This would be like a medium skin tone, which you can see from the table. Now, because of this, naturally, human genotypes can code for 7 different kinds of skin tones. The darkest being like completely homozygous dominant, and the lightest being homozygous recessive. And what we call this is you can like map all of these phenotypes on a graph almost. And because there are so many more combinations that yield for like the more middle skin tone, more people are going to be born with the middle skin tone. And if you're able to graph phenotypes like this, we call this like continuous phenotype variation, where there are so many different phenotypes that you can express, you can almost like graph them. Another example of that is hair color. Obviously, lightest hair color being blonde, darkest hair color being black. And there are about, I think it was like 10 or 11 genes that actually influence hair color. So it is a very, very, very continuous gene. We'll just put it like that. And you can graph the polygenic inheritance. Sometimes there are differences. If the inheritance is polygenic and on the sex chromosomes, the distribution will actually be different in women compared to men, which is what you see here. This is actually the height of women and men. You can see that the most common phenotype for women is slightly lower than the most common phenotype for men. And overall, male heights are a little bit more spread than female heights as well. So monogenic traits are the ones that are only coded for by one gene. Polygenic traits are the ones that are coded for by multiple. And you can plot them on a graph. So if we have a look at a practice question, this is one that came up, I think this is either 2020 or 2021. I can't quite remember exactly. To demonstrate polygenic inheritance in the color of wheat, a cross was performed between 2 intermediate parents that were heterozygous for every single one of the genes. Each dominant allele adds a unit of color to the phenotype. So what graph would depict the number of individuals and the number of phenotypes for the cross? So immediately you know that because there's 3 of them, there's going to be 7 phenotypes. You should know that from your notes. But if you didn't know that, I can teach you how to work it out. If you have 3 genes, so if it's a 3 by 3 cross, you're going to be getting, you're going to be getting either, oh my god, what is happening? Sorry, wait, there we go. You can get copies of every single capital or dominant gene. You can get a combination of 2 capitals, so A, B, B, C, lowercase c, and then alternatively A, B, B with one lowercase b, or a lowercase a, capital B, capital C. Alternatively, you can get 2 copies of a recessive gene and only one fully capital set. So you can get AA, BB, CC with lowercase b and c. A lowercase a, B lowercase b, C capital C, sorry. And then A lowercase a, B lowercase b, and B capital B, C lowercase c. Now with the phenotypes, the kinds of phenotypes that you would be expecting here are the darkest phenotype. This one would be slightly lighter because it's only got one copy of the recessive allele. This one would be lighter because it's got two copies of the recessive allele. If they had three copies of the recessive allele, so if that was A, B, B, C, C, it would be slightly lighter again. Four copies of the recessive allele, so for example, A, B, B, C, C. Five copies of the recessive allele is your next level of lightness, so A, B, B, C, C. And then finally, six copies of the recessive allele, so A, B, B, C, C would be your next stage of lightness. So all in all, we've got the darkest shade, and then 2, 3, 4, 5, 6, 7 different phenotypes. And that's where the 7 occurs on the bar graph. Now, this is the question that I was talking about, the one that came up in 2021. This is for blood group inheritance. So you've got 3 alleles, O, A, and B, with O being recessive and A and B being dominant, determining blood type. It is known that A and B are codominance inheritance, and the father who is a heterozygous for type A, so they have IA, little I, and the mother who is heterozygous for type B, so that is IB, little I, have children together. Predict the likely frequency of phenotypes for their offspring and show you're working. So in order to do a prediction for genotypes, you're required to do a Punnett square. The father I'm going to do up top, and the mother I'm going to do on the side. And draw the Punnett square. The first offspring that we get is AB, second is B little I, third is A little I, and fourth is little I. So the frequency of the phenotypes, now you've got to express them as either percentages or fractions. Both of them are equally accepted. But it's 1/4 or 25% type AB blood, because this is phenotypes, right? You want to be saying like what the trait is, not the gene. So type AB blood, 1/4 or 25% type A only, 1/4 or 25% type B, and 1/4 or 25% type O. And you get 1 mark for giving all of the Punnett square correctly, 1 mark for stating the correct frequencies, and 1 mark for making sure that you're linking the frequencies to the phenotype and not the genotype. So it's not enough for you to say like, oh, 25% IA little I. That's not a phenotype. You're not answering the question. So make sure you are answering the question to what they ask you. Another way that inheritance can be assessed is through understanding how inheritance is passed on. So describe what is meant by the genotype of an organism. This is the combination of alleles that you get from the mother and father. A gene from mother and father. That one's nice and easy. The second dot point, inherited mutations can affect the genotype of the offspring. Describe how this occurs. This is actually the third dot point in the inheritance topic. And basically, mutations that occur can only be inherited by the offspring. Can only be inherited by offspring if they are mutations in the sex chromosomes. If in the sex cells, sorry. If they are mutations in sex cells, so gametes. If the mutation helps with the survival of the organism or is not a diseased mutation with survival, it will be passed on to the offspring. It will if the mutation is, we call this deleterious or unbeneficial, as in it's a mutation that is a disease of some type or doesn't help with the offspring survival, it will not be passed on to the offspring. Onto the offspring. If passed, a new allele enters the population and can be passed on to all of the other generations. So in this one, you get 1 mark for saying that mutations can be inherited by offspring if they're in the sex cells, so here. And the second mark comes from just saying that mutations are only inherited if they are beneficial for the survival. So I will just take this opportunity to say that's the end of the inheritance topic. And so we're going to be going into biotechnology soon enough. I'll tell you back very, very soon, I promise. And if you do have any questions about inheritance, please let me know now. If not, sit back, hold on tight for some biotech. So, biotechnology is probably going to be a new topic for most of you. I don't think it's very taught in some of the previous years. And so, wait, let me go back to the slides here. So, biotech is not something that most of you are going to be very familiar with right now, but it is a very, very content heavy topic. There are 4 processes that you do need to know in pretty significant detail. And one of them is PCR. So if you've ever done a PCR COVID test, you're going to learn the process as to sort of how that happens. Biotechnology in general are any sort of technologies, machines, processes, analytical techniques that help us understand biology and human genetics in better ways. So biotech has huge applications in terms of, sometimes really simple things like diagnosing diseases, and sometimes much more significant things like curing diseases. So, for any of you doing your research investigation, if you're doing things like gene therapy, genetic screening, uh, any sort of genetic treatment, CRISPR, cloning, any kind of meddling around with genes are known as biotechnologies. So some examples can include use of penicillin as an antibiotic. Some more complicated examples are, gene therapy, where you're literally cutting out or replacing a gene in an organism to treat them of a genetic disease. There are 4 main important kinds of biotech that you guys are needed to know. The first one is how to make recombinant DNA and plasmid biotech. The second one is DNA profiling. So that's where you, it's used in forensics, where you take a sample of a DNA and then take a sample from a criminal and see if they match up. DNA sequencing. So that's where you are able to identify what the base pairs are in a certain genetic code and map out, oh, there's, this many adenines, this many thymines, etc. PCR and gel electrophoresis. So this is going to be a lot of content. I will say it's the best way to learn biotech is kind of to take it slow. So try and absorb as much as you can during this lesson. But I will say if you've got access to the recording, definitely come back to it because I'm going to kind of fast track you through 5 different kinds of analytical techniques. So hopefully I don't overwhelm you guys. But as always, I can't really slow down because you're watching a recorded lecture, but if you need some further clarification, let me know. I'll try my best to make it happen. So first one that we're going to do is recombinant DNA. Sorry, just give me 2 seconds so I can start writing. And recombinant DNA, if we want a definition of it, is taking DNA or taking a healthy gene from a different organism and inputting it into a new organism. So you might have heard like genetically modifying plants with pest resistant genes. They've taken those genes from, bacteria or other organisms that have developed a resistance to the pest and then inserted it into the plant genome. So basically, they're taking foreign DNA and putting it into an organism to make it a little bit more healthy. And basically, the foundation for recombinant DNA are these little rings of DNA that are found in bacteria called plasmids. So our plasmid here, a whole bunch of bacteria are farmed and extracted, whether plasmids or the DNA that aren't like in nucleosomes are used as sort of like transport vehicles to transport DNA into other cells. So our first step is isolation. And that is where you have something called a target piece of DNA. So this is the genetic code for the actual gene that you want to insert. So this could be like, I don't know, an antibiotic resistant gene. It could be a heat resistant gene or something like that. This is cut using something called a restriction enzyme. So this little tiny protein is engineered by researchers and cut out of the original piece of DNA. Let's say that the antibiotic resistant gene was in I don't know, a small worm or something like that. They take the worm's DNA and use the restriction enzyme to cut the code for the gene out and separate it from the rest of the organism. So restriction enzyme is used to cut the target gene um, gene. Now, the start and end of the restriction enzyme are called these recognition sites. Recognition sites, which basically identify like, oh, this is the start of the gene and this is the point where the gene finishes. There are two ways that a restriction enzyme can cut a target gene. The first one is straight down. So if you take out the little piece that it's cut, let's say that there's like, I don't know, an adenine here, thymine, guanine, thymine, cytosine, etc. And then it's flanked by GCTA as the finishing recognition site. Now, that can be cut straight down. And if it is straight, as in like the gene looks like a rectangle once it's cut, this is called a blunt end. And sometimes, like the one that I drew in red, they'll be cut in sort of this shape with like a bit of an overhang. So like the target gene will go from here to here, and then they have an extra ATG and like, I don't know, GCA at the very end. These are called sticky ends. Typically speaking, sticky ends are a little bit more favored because once we insert it into the plasmid, the plasmid will then bond to the little overhanging bits and like provide a T, A, and C, and alternatively C, G, T on the other side, so that they stick together really, really well. Kind of think of it as like piecing two Lego blocks together versus piecing two wooden blocks together, because of the overhang and because the overhang allows the pieces to fit really snug against each other, the adherence is a little bit more strong. So in general, sticky ends are preferred. Here. Anyway, step number 2 is plasmid preparation. And this is where we take that plasmid and we use the same restriction enzyme that we use to like harvest the target gene DNA to cut a little section out of the plasmid DNA as well. So the restriction enzyme, the same one. Hello? Oh, whoops. Restriction enzyme cuts the plasmid, and this then becomes a plasmid vector. The target gene, which I'm going to code in red, by the way, is then inserted into the plasmid vector, and it should fit snug against each other because they were cut using the same enzyme. If it's a sticky end, the base pairs at each of the ends will actually bond together and cause a very, very snug fit into that vector. This now becomes what is known as a plasmid vector because the target gene has been inserted inside. Step number 3 is... Oh, I don't know why it's light blue. Sorry, I wanted it to be the same blue as before. Step 3 is ligation, where the isolated DNA and the prepared plasmid are now combined. And basically, so here's our original plasmid. And on this section is the start of the target gene. On this section is the end of the target gene. DNA ligase, which is another little enzyme, another little handy enzyme, glues the two of them together to make sure they're super snug. And then finally, so once this happens, there are some of the plasmids that combine with the target gene very, very successfully. Some of them, though, don't. They just reject the target gene and end up dying off. In like real life, to check that they have been successful, what'll actually happen is an antibiotic gene will also be inserted into the plasmid vector. Is inserted. And I'll talk a little bit why that is significant in this next one. Sorry, just give me 2 seconds. Oh, there we go. Hopefully you can see me there. Oh, so now that the plasmid vector has fully been prepared, we need to test whether it's been successfully, like the target gene has been successfully inserted or not. So just as a little recap, target gene is cut from the host DNA or the foreign DNA. Same restriction enzyme is used to cut the plasmid vector, and it's combined with the plasmid. So once the DNA ligase comes in and everything's glued together, it turns into a recombinant plasmid, where it's got a foreign piece of DNA inserted into it. Then what happens is, if my PowerPoint slides are going to work, there we go. Then what happens is the bacterial chromosome will be put into a solution and heat shocked. So the temperature is like raised really, really high, and it encourages the bacteria to start growing and produce the code for its growth. What we then see is the solution of the heat shock bacteria is to be placed on a petri dish, but the petri dish is antibiotic. So the only bacteria that can grow in there successfully are the ones who took the antibiotic resistant gene from the plasmid vector. So all of the colonies that end up growing are the ones with the correct plasmid on it, and they are then harvested and then sent off as the successful plasmids to make use of the gene that's been inserted in there. So the gene then starts creating the protein from the inserted piece of the gene. So, sorry, the plasmids then start making the protein from the target gene, and those proteins are harvested by researchers who then go off to do all sorts of things. And so the final step of recombinant DNA is something called bacterial transformation, where they undergo a heat shock. The successful recombinant plasmids die. Sorry, the successful recombinant plasmids are able to grow on an antibiotic dish, and the unsuccessful ones end up dying off. Those that were successful are collected and then used for protein production. And okay. I just realized I had to flip my screen around, even though I just got it off. Or, no, I can just teach you off this. So, DNA sequencing and DNA profiling are slightly two different things. DNA sequencing is our ability to map out the base pairs of a DNA sequence, so the mixture of, G's, A's, TC's, etc., etc. DNA profiling is the ability to compare a known sample of DNA against an analyzed sample of DNA and see their differences. So DNA sequencing happens by running a sample of DNA. So, for example, here is the sorry, I'll get my laser pointer. There we go. Here is our sample of DNA that we want to copy. In order to sequence a piece of DNA, you need a lot of copies of it. Because DNA is really, really small, right? You want to get as many copies as you can so you can it becomes more of a physical substance. So the process used to copy DNA is actually PCR. We're going to go into that very, very shortly. So stay tuned for that. But the gene that you want to sequence undergoes PCR, which causes a lot of copies to come about. Then what happens is that each of the base pairs are dyed with a specific color. So guanine in this example is dyed with a yellow color, adenine is dyed with a green color, cytosine is dyed with blue, and thymine is dyed with red. And all of those dyes are fluorescent. So once exposed to a particular light frequency, they can glow. What then happens is that the sample of the DNA that is now dyed undergoes a couple of molecular processes before being run in gel electrophoresis. Gel electrophoresis we're also going to talk about in a little bit more detail, but essentially that process separates out all of the pieces of DNA so you can see each of the different components within it. The small fragments of DNA get pulled very this will make more sense when we talk about it later, but the small fragments get pulled a very small amount. Small fragments get pulled a very large amount. The large fragments travel a very small distance. So suddenly you've got all of these sections of DNA from the target gene that are now in more digestible sections. What then happens is after they are separated out, the scientists will run a laser beam through the sample of the DNA, and all of those fluorescent dyes that we were talking about there start to glow. And so you use this thing called a chromatogram, which shows the first base pair that the laser came into contact with was an adenine. And so you get this, not an adenine, let's say it's a thymine. And so you get this spike of red that comes up on the chromatogram. The scientist can read that and go, okay, it's a red dye, which means our first base pair in the sequence is a thymine. The second base pair that the laser comes into contact is also red, so it's another thymine. Then you might get a yellow, which means there's a guanine there. Another red, another thymine. Blue is next, so it's a cytosine, and so on and so forth. So the more that the laser runs through the sample of DNA, the more of the fluorescent dyes start glowing, and the more that the researchers can identify this particular color aligns with this base pair nucleotide. And then once the entire dye has been lasered through, they can put together each of the colors and find out what the base pair sequence is. So this allows us to look at exactly the sequence of base pairs in each of our gene and map out our genome. The human genome was mapped out through something called the Human Genome Project. And so we've successfully been able to get all of the sequences for all human genes, which is quite an impressive thing to do. Because of that, now we can kind of break down like, oh, this gene codes for this particular protein, which results in this disease, etc., etc. And it's really, really helpful in like diagnosing genetic diseases. So the dye process is called DNA sequencing. DNA profiling, I will come back to, but I just want to go through polymerase chain reaction first with you all. So PCR is the process that we use. Give me 2 seconds. I've got notes that I'm looking at, and the PowerPoint slides just stopped responding. Anyway, PCR is the analytical technique that you might have heard of with COVID diagnosing. Now, in order to perform that process successfully, they take a sample of your DNA that's either like a bit of your mucus from your nasal tract, or I know some people have done cheek swabs as well with saliva. There is only a very small amount of DNA in those samples that they take, and in order to correctly diagnose you of a disease, you need a much larger sample of DNA. And so the thing that lets us facilitate that is polymerase chain reaction. As you can see from the slide, the purpose of PCR is to make millions of copies of DNA sequences when only small samples of DNA are available. I've done PCR, I think, twice now, once in first year uni, once in second year uni. It takes about, I mean, you can run it for as long as you want, but we ran them for about 3 hours. And even after 3 hours, like we had this tiny little test tube that was, similar to the size and thickness of my like stylus here. And even then, the only DNA that we could see was like a small, like it was literally the size of the pen tip. There was a very, very small amount of DNA. And supposedly that was around, multiple millions of copies. The more you run it for, you can run the process for as long as you like. Within uni, we were only able to run it for 2 to 3 hours, but the more you run it for, the more copies that you can create. It doesn't really expire as a process. And so you might notice a couple of similarities between DNA PCR and DNA replication, if you remember that process from the start of Unit 4. But basically, the first thing that happens is you take your target gene or your sample of DNA that you would like to copy. So this is the target DNA sequence of interest. Now we know that in terms of DNA, there's a 5' end and a 3' end, and that DNA, sorry, whoops, that copy of DNA is then going to go through something called denaturation, which is basically a step where the sample is heated to a very, very high temperature, which causes the hydrogen bonds between the bases to break apart and then separate the two strands out. The second step that happens is called annealing. So annealing is where the temperature is then decreased. And then this enzyme called Taq polymerase, that's TAQ polymerase, comes in and starts, sorry, not Taq polymerase. What am I saying? Annealing, temperature is decreased, and then these RNA primers bind to the original copy of the DNA. So kind of like DNA replication, there's a priming step. Extension then happens where the temperature is then heated again, and then they run this enzyme called Taq polymerase into the solution. So that's TAQ polymerase that binds onto the primer molecule, so this purple strand right here, and starts copying the target gene by adding like free nucleotides. So it does the exact same thing, all of the adenines paired to thymines, guanines to cytosines, and so on. And then at the end of extension, you've gone from one copy of DNA to two copies of DNA. And then basically, immediately after this cycle finishes, it re-goes through the whole process. So denaturation happens again. The strands will separate out once again, but instead of one copy, you've got two copies being created. Annealing will happen, which causes the primers to bind onto the strands, and extension will happen where Taq polymerase goes and copies it in again. And so by the third cycle, you've got 2 to the power of 3 or 8 copies of DNA being made. And you run it through as many cycles as you need to get 2 to the power of n copies. So the PCR product that ends up happening is each time that the DNA is replicated, it gets broken apart again and re-replicated. It's quite a fast process, honestly speaking, and very, very useful in getting multiple copies of DNA quickly. Now, some useful applications of PCR, these are the ones that you're required to know. Number one is diagnosing any sort of genetic related illness. Number two, analyzing crime samples found, like either DNA or blood samples found at a crime scene, to amplify and then analyze them in forensics. Number three, potentially extract and amplify small amounts of DNA from fossilized remains. So we can look at DNA relationships between organisms. So yeah, you might have heard that humans and chimpanzees share 99% of their DNA. That was only discovered because of PCR, the ability to amplify them and then run them through tests. So PCR by itself is actually not too useful because it just creates multiple copies of DNA. In order to understand the similarities and differences between these DNAs, we use a process called gel electrophoresis. Now, if any of you are also chemistry students, you would know that gel electrophoresis is an analytical technique that's used in both bio and chemistry. But in DNA, what they do is they get this really, really jelly-like sheet. It's made out of agarose jelly, if you've ever done agar plates. And on the end of the agar plate are all of these little holes called wells. What you can do is using a pipette, you can insert a small sample of DNA into each of these wells, and then you set it into a electrophoresis chamber. The liquid that you see there just reaches the top of the well. You'll actually do this experiment. It's a mandatory practice, so you can see it in action. But basically, the liquid is set just on top of the well, and it's like a buffer solution, so it's like a salt. It's got ions in it. And then on the ends of the plate are electrodes, so a positive side and a negative side. What they basically do is they load all of these samples of DNA into each of the wells and run a current through it. And because the salt solution is able to conduct electricity, the current moves through the entire well. DNA, as a molecule, is positively charged. And so as it runs through the current, the molecule will be attracted to the negative side of the agar plate. So all of the pieces of DNA within these samples will start moving to the negative side. Once this happens, the small molecules, because they're very, very light, will travel a really great distance. And the larger molecules, because they're more dense and heavy, will travel a smaller distance, and you end up separating them out. So I've got very dense molecules on this side of the plate, and then very light molecules on this side of the plate. What they'll often do is take a sample of DNA, and put it on the first rung, and then another sample of DNA, put it on the second. The more similar bars that they have, the more likely the DNA is the same. So if this was like a crime scene, for example, and this was, the suspect's DNA that they had gotten from somewhere, and then C was the sample found at the crime scene. because those two bands are very, very different, the pattern of bands are very, very different, it's very unlikely that the suspect is the person who actually did the crime. Whereas if those bands were matching up like one for one, you've got yourself a match. And there are a lot of forensic applications that you can use gel electrophoresis for. So the uses of gel electrophoresis are determining parental testing paired with like DNA profiling. Second thing that you can do is GMO testing. So genetically modified organisms go through this process to see if their genes have been inserted properly. Sometimes you can test out champagne with gel electrophoresis and also polygenic inheritance identification. So lots of applications. Now, DNA profiling, which we saw here, is the combination of you getting a sample of DNA, running it through PCR, and then running it through a gel electrophoresis to compare a known sample against a found sample. So that's what DNA profiling is. Which brings us to this question. Now, DNA profiling came up in the 2021 exam, which is what I wanted to go through with you guys right now. It was in a short response question, so this is paper 1. If you were asked to write the 3 components of a DNA nucleotide, this one is a Unit 3 like early topic 1 question. But a nucleotide has sugar, which is deoxyribose sugar, covalently bonded to a phosphate group, and the sugar also has a nitrogenous base. Now, to get the 1 mark that this question is worth, you do not need to draw the nucleotide. You just need to say those 3 words on there. Now, describe the steps involved in DNA profiling. So DNA profiling is where you compare the similarities and differences between a known sample of DNA against an unknown sample. So for example, if you have a blood stain at a crime scene, and then you're able to get a suspect that, could have been the criminal. And then if you're able to collect a sample of blood from that suspect and compare it against the crime scene, being able to identify similarities and differences can be done through DNA profiling. The first step is to identify these things called STRs, or short tandem repeat sequences, which are bits of DNA that are common to every human. They're common genes that everybody has that repeat very, very quickly. So by identifying them, they're probably the easiest point of comparison because there are so many similarities and they're a very common type of genetic sequence. Step number 2 is to amplify the copies of DNA through PCR. And run that for a couple of hours until you've got multiple copies of it. Step number 3 is to run the amplified copies through gel electrophoresis. So gel electrophoresis and find similarities and differences. Like so. So that is DNA profiling in a nutshell. Those are the steps that are required of you to answer that question properly. And it's basically 1 mark per correct step. All right, give me just a second. Whew. All right. That is the end of our biotechnology topic in terms of content. But there is one cognitive verb that I want to run through with you guys just before we go on to evolution. One of those last dot points in the syllabus is your ability to appraise a biotechnology technique on its success rate. So the actual verb of appraise is actually very unnecessarily complicated, but there is a certain way that QCEE wants you to answer these questions if you do ever get an appraise question in your external. The first thing that you need to do is determine a measure of success. So if you were asked, appraise the ability for genetic screening to improve effectiveness of diagnosis, for example. Your measure of success might be the time taken to complete the diagnosis. It could be the accuracy of the diagnosis. It could be the severity of the diagnosis, like whether or not it's able to catch the disease at a less severe time, or whether or not it's like kind of ineffective with its method. So I've got other examples here. Growth rate and then percentage completion could also be your measures of success. Your second step is to identify a treatment group and compare it with the control to see how successful the technique is. So you take that technique that you're trying to appraise and compare it against a technique that's already used and see, are the accuracy levels higher or lower? Are the times higher or lower, etc., etc. And then finally, make a conclusion about the future use of the technique based on the success. So whether or not it's actually better than the techniques that are already used, or whether the previous one was better, so the new technique should just be scrapped entirely. And so anytime you see an appraise question, these are the 3 steps that you need to put into your question answer to get it correct. All right. Sorry, I'll just plug my charger in to the laptop. In terms of bioethics, biotechnologies are a little bit invasive because you're directly dealing with, people's genes and things like that. So there are a lot of policies put in place to protect people who are going through like biotechnology trials and things like that. These are things that you could be considering in your IA3 if you're talking like gene therapy and cloning in particular. But it's not something that will be assessed in your external. So just wanted to make sure you know that. All right, now I'm going to go into evolution. I apologize if this seems a little bit fast track. I'm running a little bit late, but that's okay. The first the dot point on there is to define evolution, macroevolution, and microevolution. So these 3 I have copied straight from the QCEE glossary. Evolution is the change in genetic composition of a population over generations, which may result in the development of a new species. So anytime a species has devolved from its common ancestor, something regarding evolution has happened. Macroevolution is like large scale evolution, where it's the variation of allele frequencies over geological time in a particular species, resulting in the divergence of its taxonomic group, where the descendant is in a different taxonomic group to the ancestor. That's how you know a new species has been formed, where the descendants are no longer in the same taxa. If you think back to classification systems, microevolution, on the other hand, is small scale allele variation, which within a species where the descendant is in the same taxonomic group. So an example of macroevolution would be the division of humans and then ape species from a common ancestor, a common mammal ancestor. Microevolution, an example, is the variation of height and skin color across the human population. Just because somebody has, blonde hair doesn't mean that they're a different species to me. It's just a difference in allele variation. So that's a microevolutionary change. In terms of what evolution is, the Earth can be mapped out according to all of these eras and all of these timelines. And because like all of these areas are characterized by different kind of environmental conditions, different kind of predator and prey relationships, and different levels of biodiversity. all of these differences cause selection pressures or things that affect a population from surviving and thriving. And sometimes there are, mass extinction events that take place. Sometimes there are sudden boosts in biodiversity. And every single time a selection pressure, affects a population, their ability to adapt to that selection pressure and change their allele frequencies and traits is what results in evolution. And so there are some important terms for you to know. The first one is mass extinctions. So mass extinctions are any periods of time where a large amount of species are dying out due to changes in environmental pressures. The species that dies out can no longer exist, so there are no species left, but there can be ancestors of these species that have evolved to become a new species. The opposite of mass extinctions are evolutionary radiation, which you can see here. And evolutionary radiation is basically, within a very short amount of time, the rapid expansion of species from a smaller number of related organisms. Evolutionary radiation can also be called adaptive radiation. They're the same thing. But basically, what you see here is differences in genetic diversity and allele frequencies can cause evolutionary radiation, which can also be influenced by things like climate change, geological change, atmospheric change, and ecological opportunities. A mass extinction is where multiple different species become extinct, and sometimes a mass extinction where all of these species are dying out opens up new niches within an ecosystem, which creates the need for evolutionary radiation. So all of these species start to divide really, really quickly, forming new species to fill up those niches that were left behind by the ones who died out previously. And so you can sort of track how these happen. So evolution is the opposite to mass extinction. Here there probably was a event that caused species to diverge really, really quickly, and this one common ancestor suddenly broke up into multiple different kinds of species within one or so eras. So that's a example of adaptive radiation there. Hopefully that is all making sense. Oh boy. And so with the eras, right, any sorts of these diagrams that we get are made by sequencing the DNA of all of these organisms. So we get samples of, fossil records, samples of organisms that we already have living, and we can compare them to see their similarity in their genetics. The more similar an organism's genetics are to one another, the more closely related that they would be. We talked a little bit about DNA sequencing. That process is what is used to do this. The official name for mapping out an organism's genome and comparing it to another organism is called DNA hybridization. And here we're extracting the DNA of two different species, cleaving it into like fragments using restriction enzymes and heating them to create hybrids with each other. The more similar base pairs exist between two DNA hybrids, the more similar those organisms were originally, and then the more related they are in the evolutionary timeline. A very common but less accurate method used is to compare organisms based on their comparative homology. So homology is literally their physical features as well. But sometimes physical structures aren't necessarily developed because of common ancestors. Sometimes it's because of common environments. So comparing genetics and DNA hybridization is always going to be more accurate than molecular homology. So with molecular homology, we can go into phylogenetic relationships. You would have come across this briefly in Unit 3 when we were looking at how to define species. But to put it simply, phylogenetic relationships are looking at how similar DNA sequences are between organisms that we know. So, comparative genomics, or comparing genomes between um, organisms, relates to all of the similarities and differences that we can see between individual gene sequences, transcription factors, the presence of duplicated genes, non-coding DNA, and repeated DNA sequences, which are the STRs that I was talking about before. If a species has a very similar set of proteins or chromosomes or DNA sequences, it's interpreted as evidence that they would have come from a common ancestor beforehand and they would have diverged or evolved differently somewhere down the timeline. Recent, though, in terms of evolutionary relationships, can mean thousands, hundreds, or even millions of years. Everything's sort of comparative to the billions and billions of years that the Earth has been around for already. So phylogenetic classification, which is what you see on the screen here, is the classification of organisms based on their evolutionary traits. And a phylogenetic tree is a diagram that shows how similarly related organisms are. So on this diagram, we have three different organisms that are being compared against each other: the camel, the peccary, and the pig. All of these are mammals. So we immediately know that they were somewhat related to each other down the line. Here you can see that the base pair sequences, part of their genome's been extracted and all of the differences have been identified. The blue bits are the similar and the black bits are the different sequences. So sometimes they can just be a base pair here and there, sometimes it's an entire codon. So those differences are then extracted and then summarized in a table. So between the camel and the peccary, there's 15 differences. Camel and the pig have 13 differences. Peccary and the pig have 6 differences. And so those can be mapped out on a diagram. Sorry. Here, where the camel was the first one to break away from the common ancestor, because it's the one with the least amount of similarity. And then the pig and the peccary are the ones that broke away much later down the line. You can either have a scaled tree, which is what you see on your left-hand side, where all of the lines kind of are proportional to the time it took to diverge. Or you can have an unscaled tree, which is a cladogram. Again, think back to Unit 3, which is just, lines showing relationships where the length of the line doesn't really matter too much. So being able to interpret some of these diagrams is what'll help you answer these kinds of questions in your external. So I have some practice questions for you now. How are we going for time? Okay, I've got about 15 minutes left, which is good. I want to see if you guys can answer this question. I'll obviously go through the answer at the end of the lesson, but because there are 4 marks on this question, this is a paper 1 question from one of the externals. Have a go at even just writing down some dot points that can be used. So you're asked to explain microevolutionary change through mutations. 4 marks means you need to be giving 4 pieces of information. So I want you to have a think, maybe for the next 2 minutes, as to what they may be. And I will go for the answer. Done. Okay, I will go through the answer now. The first thing that you need to say is that mutations happen at a natural rate. So mutations within a population occur naturally, introducing new alleles into the gene pool. New alleles into the gene pool. What this means is that these mutations can be beneficial to survival. So these alleles could increase the rates of survival compared to the existing gene pool. They could be neutral, so they have no effect on survival, or decrease survival chances. If there is an selection pressure, that a new allele can work to improve. Oh, oops. Oh, wait, did that just get rid of my... No, okay. I'll just go here. I'll paraphrase. Sorry. Mutations increase... Oh, mutations are natural and they increase the number of alleles. They can be beneficial, detrimental to survival, or neutral. If there is an environmental pressure, a new allele is beneficial against selection pressure. There is an increased chance of offspring being born with this allele. If this happens over, multiple generations, this continues to occur over time. The new allele's frequency will increase. And existing allele frequencies will decrease. Will decrease, causing microevolutionary change. And you basically get 1 mark for talking about the natural rates of mutations, 1 mark for saying that it makes more alleles which can have, beneficial, detrimental, or neutral effects. 1 mark for saying how the allele frequencies can change over time if the new allele helps with the current environment. And finally, that change in allele frequency is what we call a microevolutionary change. All right. Explain a possible consequence of a population bottleneck. All right. Couple of things here. I need to go through the theory for you here. Population bottlenecks kind of align with mass extinctions, where it's the dying off of a species within a very short amount of time, of multiple species, species in a very short amount of time. So, for example, if you had an ecosystem that had 57 species, and then there was a natural disaster, like a forest fire, that wiped out until there were only 10 resulting, this rapid decrease is a population bottleneck. And it happens in a very short amount of time because of a natural disaster. And sometimes the surviving population, surviving population can be representative of the original population. But oftentimes, the surviving population is a lot more vulnerable. And I'll just explain why. If I go back to my example that I've given you of the 57 original species that existed here, let's say that 20 of these species were producers, 17 of these species were herbivores, and 15 of these species were small predators, and the remaining 15 were higher order predators or large predators. Let's say in the surviving population, we could have had one producer, 3 herbivores, 3 higher order predators, small predators, and 3 higher order predators. Or we could have had 10 higher order predators as the remaining population after this natural disaster. One of the species has one kind of organism from each of the main niches. There's like something that can produce the food, something that can eat the producer, and then a couple of predators to eat the herbivores, prey species. In the second case, where all of the surviving organisms are predators that need prey to survive, that second scenario has the organisms kind of struggling to survive. There's no diversity. And the reason why this happened is because, yes, because of a natural disaster, but sometimes it's completely random. There's not enough time in one of these like severe natural disasters for natural selection to take place and the survival of the fittest to occur. The resulting populations can either be like super representative of the original, or they can be a really, really bad representation of the original population. So, population bottlenecks, if I was to answer this question, population bottlenecks occur during um mm, oh sorry like rapid natural disasters, often resulting in an extremely reduced vulnerable surviving population. Because of the decreased biodiversity, this population is at risk of because of the decreased biodiversity, the environmental pressures take a greater effect because we don't know if they're ready to adapt to this environment or not. Take a great effect, meaning they must quickly adapt or go extinct. So that's one of the consequences. They adapt really quickly and undergo adaptive radiation or go extinct because they're unable to cope with the environment. Obviously, the more representative the population is, the more likely that the adaptive radiation will be successful. And the more vulnerable a population is, the more likely that they're going to go extinct, unfortunately. All right, my final question for you guys is one with a little graph here. So this graph is showing you the number of families in an ecosystem across all of the geological time periods. So your question is to state which geological period reptiles first appeared. So reptiles refer to this like gray color here. And if I trace it back to the origins, they kind of originate at this point, which is in the Carboniferous period. And you get 1 mark for saying that. If you wanted to calculate how many families of mammal were present 5 million years ago, 5 million years ago is the very, very end of the graph. And so we're wanting to track how many mammals first started. So that comes around 200 to about 325. So the total amount of families occupied by the mammals was 125 families. Infer when a mass extinction occurred. So we're looking for like rapid decreases in biodiversity. The first one I would probably say here at the very end of the Cretaceous period, or alternatively at the end of the Permian era, but the Permian one was slower than the Cretaceous period. So mass extinction at the end of the Permian era was not as significant as the mass extinction of the Cretaceous period. And so either at the end of the Permian era or the Cretaceous area. And the reason why we know this is because of the sudden drop in families and species. Alrighty. That is all of the content that I have for you guys today. Let me just put the camera back on and you'll be able to see me again. A transcript of all of the notes that I used about biotechnology, about inheritance patterns, and about evolution and adaptive radiation and all of that jazz is, there we go, sorry, is available to you if you scan this QR code. And as always, if you have any further questions, I'll be sticking around for a little bit after the lecture to answer them. Other than that, best of luck for all of your studies. Sorry, I'll keep this on. Best of luck for all of your studies. Hopefully, the external is kind to you guys this year. And if you've got any other internals left, good luck with completing those. Other than that, I hope to see you at the September lecture series, and Hopefully you can also check out some of the other lectures that are available in this round of holiday lectures by us. So yeah, thank you everyone, and I'll see you next holidays.